Good afternoon, everybody. Happy to be with you today. We have a great speaker without any more delays. This is Petra Proitsi from Queen Mary, University of London. Great work on Alzheimer's, nutrition, and other risk factors, epidemiological and human studies. I don't want to take more of your time. Petra, thank you very much for joining us today, for accepting to join us, the HIAAD Bridge seminars. And I hope that people will enjoy your talk and it will be a great discussion. I know your work is fantastic. And once more, thank you for joining us. The floor is yours.
Thank you so much, Ioannis. Good afternoon or evening, everyone. My name is Petra. Ioannis introduced me, and I'm based at Queen Mary, University of London. And I work on Alzheimer's disease and more recently on Parkinson's disease as well. Shall I share my screen? Shall I start sharing? So let me see. Oh, okay. Can you see?
Yes.
Yes, good. Okay. So yes, I'm based at the Center for Preventive Neurology at Queen Mary, University of London, and our research focus in a glance is brain health and neurodegeneration, where we are combining different statistical approaches, such as from machine learning methods for to improve early detection. For example, as I'm going to go through some of our work, we try to understand, to combine lifestyle information and lifestyle factors and molecular biological layers to understand more about, to improve early prediction of Alzheimer's disease and Parkinson's disease. And also one of the other themes focuses in our work is using causal inference approaches to understand more about etiology. So we work a lot with lifestyle data throughout the life course, and we work a lot with genetic and multi-omic data. And mainly today I'm going to focus on proteomic and metabolomic data, but obviously we work on the full spectrum of multi-omic data. So I'm going to give some example of some of the work we've been doing in the last, I suppose, couple of years. A flavor of a few projects, and then please ask me any questions you want at the end. I've got some additional slides of some other work we've been doing, and it would be great if I have any feedback or any ideas for collaborations and so on. So the first work I'm going to describe is focused on inflammatory blood proteins and risk of dementia. So we work a lot with local data sets, but also we work, as many people, I suppose, with the UK Biobank, with all of its pros and its cons. And what we're trying to understand is whether we can find signatures that are associated with risk of dementia, but also understand more about, as I said, molecular pathways and causal pathways. So the first study I would like to talk about is whether inflammatory blood proteins are associated with incident dementia. And then this is a work that's conducted that we've published a couple of months ago about our PhD student Dorsa. So the questions we wanted to answer is whether actually inflammatory proteins involved in inflammatory processes are associated with incident dementia. Whether those proteins are associated with image-derived endophenotypes, so we looked into brain imaging in healthy participants, and whether they mediate the association between genetic and modifiable risk factors with dementia. And then finally, whether any of these associations could potentially be causal through causal inference approaches. And the way we've done this first piece of work, and this is actually two studies that were run in parallel, so they're using the similar pipeline, is that we used the UK Biobank, 500,000 individuals, and we used the inflammatory panels, about 728 proteins from the inflammatory panels. And then we performed a machine learning pipeline that I'm going to go through in not so much detail to identify a signature, so a combination of proteins that in people before the onset of dementia. So everybody, when we started the study, we excluded everybody with anyone with prevalent dementia. And then we want to see whether our signature predicts future incidence of dementia, and then followed by associations with imaging endophenotypes and brain age, as well as mediation analysis. So this is just for those who are interested in the pipeline we've used. So what we did is we split the UK Biobank in 3 data sets, 50%, 25, and 25, and we used a regularized Cox proportional hazard models with LASSO. So we tried to train a model to find us a combination or a sparse combination of proteins that can predict future dementia. So what we did is we first of all we wanted to estimate our parameters, and then we went to 25% of this data set to try to understand, based on that we've set aside, whether we can actually try to find the features that are most important for our model. And then in the end we went to our test data set that we kept aside again, and we tried to see whether this signature can actually improve prediction more than traditional clinical risk factors. And the way we actually did it is, the way the model works is that you have different solutions, and what we did is that we decided that this is a solution, a model solution just with one protein, and this is, you see, ApoE, and then in the end it would have been 700 proteins, and then we compared different signatures with different number of proteins, and when we had a plateau, so adding actually more, making our model more complex, doesn't explain any more of our prediction, we stopped. And we compared these models to see that statistically, by adding any more proteins, we don't improve our prediction. And this was a model with, I think, 19 or 20 proteins. And what we saw is that it did better than age and sex, and it did better than ApoE, and it did better than adding lifestyle information, but it did not do much better when you add lifestyle information and ApoE together. And then what we wanted to see is that, okay, we've got these proteins which look interesting. Can we find out more about this protein? So we did a series of regression analysis to try first to understand whether we can see any associations with different brain regions, as well as white matter hyperintensities. And brain age. So trying to understand, like, the brain age calculated as the difference between, your predicted from imaging between your chronological age and your brain age delta is the difference between your predicted imaging age. And we did find some interesting associations. What I want to say mostly is that we find 2 proteins that seem to be associated with both dementia and subcortical brain volumes, including the hippocampus. And we've previously, one of these proteins was TNFRS-F11 beta, and we've previously actually shown that this protein is associated with Alzheimer's disease and CSF and PET amyloid, as well as CSF PET and tau. So the next thing we wanted to see is whether any of these proteins that seem to be associated with incident dementia could potentially mediating the role of modifiable risk factors with dementia. So we looked in the Lancet Commission, in famous 14 risk factors, and what we saw is that some of these proteins could potentially mediate the effects of some of the risk factors with dementia. We actually looked more than the modifiable risk factors. We looked at a collection of risk factors. And for example, we saw that TNF-RS-F11 beta could potentially mediate the association of smoking with dementia. Obviously, these associations are observational, so it's very difficult to disentangle cause and effect especially when you've got cross-sectional smoking or protein data, but also they give us an idea for future analysis. When we actually performed Mendelian randomization analysis, which I'm going to talk a little bit more later, a method that uses genetic data to proxy a lifestyle or molecular data to try to tease out cause and effect, we did not find evidence for a causal association between these proteins, but nevertheless, our sort of conclusion from this piece of work were that these inflammatory proteins were quite good predictors of incident dementia, and they are associated with brain endophenotypes and lifestyle risk factors. So the next thing we wanted to do, so this is another study that was taken at the same time in parallel place. We are interested also in the relationship between different dietary scores and dementia. So at the same time, we tried to look whether, and although, as I said, the diet is not part of the Dementia Lancet Commission, we know that diet is very important in dementia, and also we know that this is not an established association because it's very difficult to study diet. So we are not dietitians. We don't work in nutrition, but we are interested in the role, obviously, because you're looking into proteins and metabolites as well. We are interested into the relationship between dietary factors and nutrition with dementia. So what we did is that we went back to the UK Biobank and we calculated different scores. So we calculated MIDAS and MIDAS C, pyramid, the alternate Mediterranean diet, DASH, MIND, that people may be familiar, the alternative healthy eating index, and the healthy plant-based diet index, because we've done this work about a year and a half ago, that there was a Nature Medicine paper looking at different outcomes with connected to these diets, and cognition was they were associated with cognitive performance, quite a lot of them. And then we know that many of these scores have been associated with dementia. So what we wanted to see first was whether any of these diet scores are by themselves associated with dementia and brain health. The association of dementia with dementia, we knew, it's been reported before, it's been published before. So, but what we did, what we wanted to see is whether they were associated also with brain age and different imaging phenotypes. So what we did is a series of regression analysis to try to understand whether any of these diets are associated with all-cause dementia. So, and just to say that before I said dementia in the previous study, we used all-cause dementia for our analysis. But here we split we investigated into more depth whether individuals having Alzheimer's disease and vascular dementia. So what we wanted to see here, what we saw here is lots of the scores, for example, particularly MIDAS, pyramid, and MIND were associated with all-cause dementia and vascular dementia, not so much with AD in this subset of people from the UK Biobank. But the other thing we saw, which was very interesting, is that, as and not unexpected, is that many of these scores were associated, for example, with white matter hyperintensities. And some subcortical volumes as well. So then what we did next is that we took the scores that were associated with all-cause dementia and Alzheimer's disease, and we looked whether any of these association, in the same way we did before with our mediation analysis, are potentially mediated by the inflammatory proteins that we found in our signature. And what we saw consistently is that yes, they are, and the associations we saw is that actually, first of all, diet is associated with changes into these inflammatory, lower levels of these inflammatory proteins. So higher levels of, for example, Mediterranean or the pyramid score or DAS diet are associated with lower in levels of these inflammatory proteins, which is very interesting. And then when we went to see whether they potentially mediate any of these proteins, the association, again, we saw some interesting associations. For example, we saw that some of these proteins, such as the one we previously singled out, TNFRS F11B, could potentially mediate the effect of MIND diet on risk of all-cause dementia, and other proteins as well. Again, these associations are, we cannot establish at this stage cause and effect, but they're quite interesting to follow up. And in fact, we are actually looking at trying to find different data sets to replicate them and study them in more depth. So the next study, at the same time we're doing this, in my group, we work a lot also with metabolites, blood metabolites and lipids. And we've published different studies on using different platforms from in-house mass spec lipidomic studies through our collaborators at King's College London to using a metabolome mass spec. And in this case, what I'm going to talk a little bit about is the NMR metabolite data from, again, the UK Biobank. So we know that lipid metabolism is implicated in Alzheimer's disease. We know that the brain is enriched in lipids. We know that ApoE and other lipid genes are very important in dementia. And we also know that lots of these small lipids especially can cross the blood-brain barrier. So in a very similar way, so these studies were sort of going in parallel. We asked the 3 following questions: whether there's any signatures that can predict dementia, whether they are related to brain imaging phenotypes in healthy participants, and whether any of these metabolites mediate the relationship between known risk factors and dementia. And this work is currently under review, and there's a preprint for it. So the pipeline was very similar, but now we had more people. So because the UK Biobank at the time, now everybody has this data, all of the 500,000 participants, and you are actively replicating it. But when we did the study, about 250,000 participants with no prevalent dementia at baseline had metabolomic data. And again, we did a similar pipeline as before. So just to say that the Nightingale NMR metabolites measure different lipoproteins, subclasses, as well as fatty acids, amino acids, and some ketone bodies and glycolysis related metabolites. So the first thing we saw in this analysis, because we had more power, we split, we looked into all-cause dementia, but then we also looked at Alzheimer's disease and vascular dementia. So the first thing we saw is that a large number, which we knew already from other published studies, was associated with vascular dementia, more than vascular dementia than Alzheimer's and all-cause dementia, something that is expected. This number decreased actually once we adjusted for ApoE carrier status, and then once we adjusted for modifiable risk factors, and even more so for vascular dementia, where suppose lifestyle modifiable risk factors such as hypertension, cardiovascular disease, and so on are probably more strongly associated with vascular dementia. I'm gonna jump the next slide just for the interest of time. I just wanted to say that the reason we are doing that is important that we know that lipid metabolism is important in dementia, but it's not only about cholesterol and HDL and triglycerides that you probably know better than I do. It's, lipid composition of lipoprotein subclasses can actually help us understand more about mechanisms. The other thing we did, again, I'm not gonna, I'm just putting this information out there in case people are interested, and it can also be the beginning of new collaborations, is that because we've done some work on ApoE and sex specific associations, so we splitted our sample, we did interaction analysis, and then we stratified our samples, and we saw that ApoE4 status and biological sex modify the associations between some of these metabolites, particularly some fatty acids and dementia. And I don't have it in these slides here, but last year we published a paper on lipids, phospholipids and dementia, and we saw that Alzheimer's disease specifically, these associations were more pronounced in females. So this kind of work also supports some of our findings. So in this analysis now, the model is exactly the same. We use a regularized Cox proportional hazard model. We did parameter estimation, selection, and evaluation. And again, we chose the best a solution that explains that predicts incident dementia in our sample, and then we found we've got 3 different signatures here: one for all-cause dementia, one for vascular dementia, and one for Alzheimer's disease, of overlapping but some different proteins. And then when we try to see whether using this signature predicts Alzheimer's disease or vascular dementia or dementias in general better than modifiable risk factors and ApoE, what we saw is that they do. But obviously, once we add ApoE again, this association is just about significant for Alzheimer's and all-cause dementia, and not significant for vascular dementia. The other thing that was interesting is that, so this is a score of different proteins that if you, so imagine is somebody having a score of their summed up effect of these proteins, the more of this score you have, the more you can predict of your future risk. So we noticed the dose-response relationship between this score and the risk of dementia. And looking into brain imaging phenotypes, we saw some stronger associations than we saw with inflammation, with the proteomic study. Of course, here we've got 5 times the number of people involved. And also we know that some of these proteins, some of these metabolites are implicated in dementias. So what we saw is that, for example, some of the things I will talk in the rest of the talk is protective associations for the ratio of linolenic acid and glutamine with with brain age and different endophenotypes, and we saw that the broadest pattern of relationships, if you look here on the right, is shown with vascular dementia. So most of the metabolites that made up the vascular dementia risk score showed most of the associations with the regional volumes, which again is something that we probably expected. We then went on to different samples. So we looked at ADNI, that obviously you are probably very familiar with, and a local data set. We've got the AdNeuromed and Dementia Case Register, and we managed to replicate some of the associations we found with Alzheimer's disease controls and MCI. And the other thing we did is we looked also at some of the proteins, some of the proteins involved in neurodegeneration and dementia, such as GFAP, NFL, and p-tau217. And again, we saw some interesting associations, especially, for example, you see here with leucine, which are actually quite consistent across the different cohorts. Then the next step we did is that we wanted to see again, do these proteins mediate the potential association between any of the modifiable risk factors with any of these dementias? And again, we used a series of mediation models. And what we saw is that consistently some of these metabolites are associated with, can potentially mediate the effect of modifiable, particularly cardiovascular risk factors with dementia. And we sort of singled out here the ratio of linoleic acid, which is to total fatty acid ratio. So it's a polyunsaturated fatty acid, because we saw most of the associations with it. And then when we wanted to see whether any of these metabolites are associated with any of the diet scores, we saw a very broad pattern. We saw that most of the metabolites were associated with most of the Mediterranean and diet scores, and actually most of the scores, in fact. So adherence to most diets was associated with a lower metabolite risk scores overall. And adherence to most diets was associated with lower metabolites, individual metabolite levels in over half of them, of the metabolites, which is very interesting. And then the next thing we wanted to see is whether we can tease out a little bit cause and effect. Obviously, this is all observational data. We don't work in the lab. We're using statistical approaches to try to understand more about these relationships. And one of the methods we've been using that has a lot of attention, positive and negative, in maybe equal measures sometimes, is Mendelian randomization. So basically, we are using genetics as instruments to proxy the relationship of modifiable risk factors with our outcomes. And I can talk about that a bit more later, but I will not go into more details for the sake of it. But the idea of it is that if you're using your genes as proxies, you overcome problems of reverse causation, because your genes are at the start of the processes. So you overcome problems with reverse causation. Also, you can overcome some problems with confounding. So what we saw here is an interesting picture that was a little bit more mixed. Some of our most interesting findings is that we observed now a causal relationship between linoleic acid and dementia, but in the opposite direction to what we saw observationally. And the same thing with glutamine. I think these are some of our most interesting associations. So glutamine genetically proxied levels of glutamine seem to be associated to be protective for Alzheimer's disease. And because we did not have a lot of power for vascular dementia, we used white matter atrophy as a proxy. And in this case, we saw that the percentage of linolenic acid, again, genetically proxied, is associated with increased white matter atrophy. So what we do in this case, so what we do in this kind of analysis is that we generate a score, but now this is based on your genetic variants associated with your metabolite of interest. So in what we saw in sort of in short, is that these relationships were actually really influenced by a specific genetic variant, which sits at the fatty acid desaturate cluster locus. And this is something we've seen before. We've previously reported that in a study I'm going to mention briefly before. And lots of people have identified associations. So this is one of the most pleiotropic loci, I suppose, ever, and it's associated with obviously lipid metabolism, but also it's associated with depression. It's one of the depression loci. So what we saw is that that particular variant was driving the association between the genetically proxy levels of linolenic acid with white matter atrophy. So then we did something, we did some statistical colocalization analysis. We wanted to go and focus in that locus and see, okay, is it the same locus that is associated with linolenic acid, the same causal variants associated with linoleic acid levels that are associated with white matter atrophy? And the answer to that was yes, it is. So, and the next question we asked, okay, what if we looked at gene expression levels? Is that locus associated with expression changes in genes in this region? And the answer is yes. And then do they colocalize? And the answer was yes. So gene expression at the FADS1 locus colocalized with both linolenic acid and white matter atrophies with strong probability. This means that the same causal variant is associated probably with all three phenotypes. And then our final step was, okay, could metabolite levels be associated with white matter atrophy? And is there a chance they mediate the effect of gene expression levels on white matter atrophy? And then the answer to that is potentially yes. We see a small proportion of mediation, but statistically significant, that the expression of this gene is associated with white matter atrophy levels via changes in these metabolites. So the next thing we did is that obviously this specific fatty acid was chosen because of our prediction model. So out of the 250 or 300 metabolites, our prediction model gave us 15 that better predicted dementia. What if we go back and we look at other polyunsaturated fatty acids? And the results that were even more interesting, similarly interesting, and not unexpected, given the literature, is that we saw that DHA and omega-3s are associated again with white matter atrophy, and again is the same variant that is driving these associations. And the nice thing I think with this is that these were more consistent with both observational data and genetically proxied data. And then this just highlights again the importance, something that it's been in the literature for many, many years, of diet and polyunsaturated fatty acids in dementia. The other interesting association we had that we had previously published twice, more recently in our work using a different metabolite platform, Metabolon, is the association between glutamine and Alzheimer's disease. So again, what we saw here is that this association is robustly protective when you're using a genetic instrument, and it's driven mainly, but not only, by a variant very near the glutaminase 2 gene, which is very interesting. And actually, after we submitted our preprint, the biggest and more recent preprint of Alzheimer's came out, genome-wide analysis of Alzheimer's, and now they nominated that locus as an Alzheimer's disease locus. And we think that's really, really interesting, because if you colocalize glutamine levels with Alzheimer's disease, we saw again that it's the same variant that's associated between Alzheimer's disease and glutamine levels. And then again, when we try to understand what's happening with gene expression levels, we offered that not the glutaminase gene actually, but I think that biologically plausible is the glutaminase gene, glutaminase 2. We saw that statistically colocalized another gene in the region SPRITE4, where it and what we noticed in this case is that glutamine mediates the effect of that gene on Alzheimer's disease levels. So basically, gene expression levels on that gene are associated with Alzheimer's disease, and this is mediated via changes in glutamine levels. And obviously, all of this again is through Mendelian randomization analysis. So we are using again genetic instruments as proxies. And the final interesting thing, again, that we had already seen, but now we see it in much larger scale, is that what we like to do, as you obviously, I did not specifically say it in the study, but obviously it's alluded, is we like to link modifiable risk factors with molecular data. We try to understand whether any metabolites or proteins, as I said, mediate the effect of any socioeconomic or specifically cardiovascular, cardiometabolic risk factor on Alzheimer's risk. And then what we saw is that glutamine potentially mediates a small proportion of educational attainment with Alzheimer's disease. And quickly, just to say here that these are all, this is very, very busy, and I just stole it from one of postdocs, Lachlan. This is work we published the year before, looking at shared metabolomic risk factors between 4 psychiatric and 4 neurological disorders. So we looked into Alzheimer's, Parkinson's, ALS, MS, and then schizophrenia, depression, bipolar disorder, and anxiety. And what came out, and we used a different, we used Mendelian randomization, and we used a different panel for metabolites, which is based on is based on mass spec. So it's completely different metabolites, but we observed again associations between glutamine levels and Alzheimer's disease. And we also observed lots of associations between fatty acid and some of the other neuropsychiatric symptoms. And I'm just putting that there because actually we haven't really followed that up so much, but this is very interesting work. We see acetylcarnitines, phosphatidylcholines, and polyunsaturated fatty acids associated with different neuropsychiatric disorders. And specifically, for psychiatric disorders, sphingolipids and polyunsaturated fatty acids came through. Another piece of work I'd like to sort of showcase that we've been doing. This is again using the UK Biobank. That's very recent work that we haven't published yet. We're just writing it up. Is whether we can actually predict again future dementia up to 50 years before clinical diagnosis, but whether there's different signatures between females and males carrying and not carrying ApoE4. And this is work done by our PhD student, Sarah. So she used again the UK Biobank. And now what Sarah did is she actually used NMR metabolomics and proteomics together. And she used demographic and modifiable risk factor together. And her outcome in this case, because of small numbers, is all-cause dementia. And she did a similar but slightly different pipeline where she's used XGBoost, and she looked at the outcome as a classification problem, not as a sort of survival problem. So she used at the end of this period who actually had dementia or not, and then looked into biological interpretation of the molecules. And I'm not going to talk so much about that, but I think, again, this is worth mentioning because what we saw that's very interesting is that we saw different signatures of molecules, some shared and some distinct between female and male carriers, particularly, and also between ApoE4 carriers and non-carriers. So the top predictors were the same across ApoE. No, there was substantial heterogeneity, or the opposite, in the top predictors across the different strata. And what we saw, for example, is that although proteins dominate these models, because proteins are very predictive of dementia, we saw that lots of the metabolites were actually predictive of dementia in women, specifically women with ApoE, which I think is very interesting. And then we saw that there's different when we looked into biological pathways, we saw that there's actually different patterns associated with in female carriers versus male carriers. And this is something now, so this work is based on the UK Biobank, but Sarah has replicated some of the work because in the GMPC, but this is the proteomics work because they don't have metabolite data yet. And then I speak very fast, super fast, so I hope I make sense. But I'm going to finish with a last slide. There's other things we do. As I said, we work in Parkinson's a lot as well. So we've got more projects starting on Parkinson's disease. But the other thing we're doing is we don't work only with blood. So another recent paper work we did actually started many years ago, but for various reasons we just completed now, is to look at different lipids in brain tissue as well. So we published this about 6 months ago. We had a collection of post-mortem brain samples. So we had about 47 people that had hippocampal volume donors from hippocampus, and then 55 from BA9. So the 47 had both, they're overlapping. It's not different people, but we just had more people with BA9. And so what we did, we sequenced the brains, tissue from the brains for TREM2 rare variants, and we found 6 pathogenic mutations. And then we did brain lipidomics this is with my colleagues at King's College London, and we annotated 107 lipids through mass spectrometry. And what then we did is we used network analysis, so we used WGCNA to try to identify whether there are any clusters or modules of lipids which are associated with Alzheimer's disease and with TREM2 levels, not TREM2 levels, so with TREM2 carriage. So the results were quite interesting. So we find a module that is called the turquoise module, because the color is completely random by chosen by the programmers. People who work with it would know. The interesting thing is that we found a cluster or module of lipids which was enriched in ceramides and sphingomyelins and phosphatidylserines. And we saw that mostly in the BA9, as you see here on the left, but also a little bit on the hippocampus, but we have smaller sample size. So what we saw here that was extremely interesting is that, for example, I've got on the screen our most statistically significant ceramide. What we saw is that, and on the right, the phosphatidylserine, their levels were increased in Alzheimer's disease. As you see here, overall in the violin plot, we've got on the left controls and on the right Alzheimer's disease. But what was even more interesting is that if you are an Alzheimer's donor and you carry one of the TREM2 variants, the levels of these lipids were even higher. We saw almost a linear effect going from control to Alzheimer's disease to TREM2 for these different molecules. We know that TREM2 affects lipid metabolism, and we know it binds to phospholipids and phosphatidylserine. So we think that these findings are quite interesting, and again linking genetic variation to lipid levels in Alzheimer's disease. So some of the key takeaway messages from this very fast and I hope not very incoherent talk is that through our work, we see that metabolite and different proteins, specifically looked into inflammatory signatures, can predict dementia and dementia subtypes even beyond some conventional risk factors. And we see some sex and ApoE specific relationships. I want to say that the inflammation work, we've got a project now looking at the links between cardiovascular disease and dementia, and we see that lots of these proteins are also associated with cardiovascular disease, and we are also looking into heart imaging. And we to try to understand a bit cardiac remodeling and brain structure, and this is an ongoing work, but it's actually results are looking very interesting. We see that lots of these proteins or metabolites are associated with imaging phenotypes in healthy individuals. So none of the people with imaging had Alzheimer's or vascular dementia at the time of imaging. And we see that potentially they could mediate the effects of some of the lifestyle factors with Alzheimer's disease and vascular dementia. And our causal inference analysis again highlights some metabolites as potential causal candidates for further study. And we're very interested in the association of polyunsaturated fatty acids and linolenic acid, polysaturated fatty acids and glutamine in these relationships. And then this work was done by some amazing people in my team. Mostly the results I've presented are by Dorsa and Yue, and Sarah should have had her name here in and Lachlan in bold, and with lots of collaborators across Queen Mary, King's College, London, and other universities. And thank you for your attendance. And I'd be happy to answer any questions or discuss further.
Thank you, Petra. It was really nice work, a lot of work. You can stop sharing. The floor is open to discussion. I think there would be plenty of things that I can imagine you can done. I don't know if Kostas or any other of the clinicians you have any question, but definitely the pipeline of analysis you have been established in Queen Mary, University of London. It's absolutely relevant of the work that, or the clinical studies that we run in collaboration between different members of the HIAAD. Professor Kostas Leketsios is here. He can tell you more about the ginger and all the other stuff that where we have biological, but also we have, trying to characterize cohorts in Greece. Costa, you have any addition? Would you like to add something here, Petra?
Well, very nice talk. I'm still catching up. You're right. You speak a little fast, so I'm still catching up. But I'm wondering if you can pull this together all, because you have lots of different methods, lots of different studies, lots of different associational findings. Can you pull this together in a theory about one or more types of dementia that you would be able to predict based on your pre-dementia findings?
Thank you. I would say mostly towards vascular dementia. I think most of the associations, especially with the metabolites, point towards vascular changes. I would say the two things we see is some of the associations we see, for example, with amino acids are more specific to Alzheimer's, and all of the lipid analyses are sort of pointing more towards vascular dementia. Another thing I'd say that we are very interested in doing, and we're sort of hoping to start doing now, is look into more pathologies in detail, moving on from the clinical diagnosis into the pathology, the specific pathologies people have, because we know that the diagnoses are not so accurate in the UK Biobank on some of these studies. But overall, I would say that our lipid work is pointing towards vascular dementia and vascular changes we see from the white matter hyperintensities, and some of the associations we see with glutamine, with some amino acids, are mostly related to Alzheimer's disease.
Well, let me push you a little bit more in that direction because you really do need to get into pathology. Vascular dementia is recognized as a clinical entity, but very rarely occurs alone in pathology. It usually co-occurs with Alzheimer's. And it's the co-occurrence, of course, that has been a big challenge. So again, is there a theory you have, even for vascular dementia, where you're trying to pull everything together or some subset of all your associations that would help us mechanistically?
A theory? No, I don't have a theory yet, but what we are trying to see, I think, is what we're trying to do right now is try to get hold of different brains with different pathologies and try to phenotype some of these molecules in these molecular layers to try to understand what happens more at molecular level. This is what we're trying currently to do. With the theory would be that, some of these associations are, they are co-occurring, they are not happening in isolation. And some of these changes would probably reflect these mixed pathologies. But I haven't got any deeper than that, but this is where the direction we are looking.
Okay, Petra, trying to connect with the clinical studies or the data we have in different members of HIAAD, because we see your pipeline, we see that works with thousands of data, but if we go down to, let's say, like 500 to 800 cohorts, would be okay for you to, and what type of data you prefer more with for smaller amount of samples of patients or, healthy volunteers?
Absolutely. I mean, some of the studies we have are quite smaller as well. So, this work now is mostly on the UK Biobank, yes, but lots of we work with others, much smaller cohorts. I think then the question would be to have a specific hypothesis and not to do like
definitely,
an agnostic analysis. But lots of these pipelines, would work the same. And I mean, some of these methods are just regressions that just take into consideration, for example, the correlation between the features you put in. So they would definitely work with smaller numbers.
Okay. Now, we are also focusing more and more in different projects in more AI or machine learning driven platforms to analyze big data. How would you consider your work, or what have you been using in Queen Mary on that big data analysis? Are you using all your own platforms, or you have internal analysis that people could also, through collaboration with you, use it? Or you think, because correlations, and as Costa said, correlation associations are good, very informative, but we are looking, but the next step is also to look for the driving force of this change. Let's speak about the nutrition to sugar, whatever, that has a specific footprint on the multi-omic profile on these patients. But what is the driving force? Was this 2-3 signaling or molecules, or something that can be the driving force of the whole changes? I think this should be something that an AI and machine learning model on your data could give us.
Yeah, I mean, we don't use any methods that are, like that. We are using methods that lots of other people are using, to be honest. So we're using simple machine learning, and especially for smaller data sets, simple machine learning methods. We started a project on using foundation models right now, to which I cannot claim expertise, but we've got a student working on foundation models, which, then you actually try to understand a little bit of, it's not a black box, and you try to you see a little bit more what's happening. But we are using methods, machine learning and deep learning, or using R and Python. And then the other thing to understand more, because my background is mostly on genetic epidemiology, is to trying to link with genes. So for me, is always trying to anchor whatever molecule we have to genetic variation and sort of use that as our anchor to find QTLs, quantitative trait loci associated with that, and then go from there. So for the sort of causality question, if I understand your question, we are sort of using genetic epidemiology approaches to try to unpick this. And then for the models, we are using machine and deep learning models, and then started now looking into more like large language models. But our pipelines are quite standard, I would say.
Okay. I mean, some of us have used the HIAAD Bio, which is a startup from University of Crete.
Okay.
And which work
I don't know
with different data. HIAAD Bio, it's GAD Bio. I mean, other also, but I've seen a lot of publications from cancer and from different brain stuff and metabolomics data, multi-omic data, including MRI. And I think Marius is in the field to also tell you more, because he's the expert from University of Ioannis University from Corfu. But in general, that's the idea of trying to combine, and they have two nice cohorts, and including the ginger now, the new study that HIAAD and Panos Alexopoulos unfortunately cannot be here today because he's in a conference. Again, through the HIAAD study in Greece, I think that will be a lot of opportunities that you can interact using data and different people in the HIAAD here.
That would be amazing. I've heard of the HIAAD study and the ginger study. I don't know so much about them, yes, but
Mari, you want to add something?
Yes, it's very, very nice question, Yanni. And the point here is if the data are relative quantification or accurate quantification, I think. And I think I saw that in your initial slide, you used 50% of your data for the training and 25% for the test, as I remember. Yes. But the question is, if these data are absolute quantification or relative quantification, because we are working also with Vaso Maherakin and similar project with lipidomics, and the critical question is this one: if the initial data set starts from absolute quantification about that, but in order, you have better training sets. This is one point of question.
It depends on the data. So some of the methods are absolute and some of them are relative. So it depends on the platform. But we've never, we've just worked with the data we had, I suppose. But yes, lots of it is relative quantification, though. Yes,
Okay. And another question about more molecular. You focus on this desaturase enzyme, which convert the DGLA to arachidonic, as I remember.
Yes.
Have you ever tested other enzymes, for example, the delta 15 desaturase will convert, omega-6 to omega-3? In order to find out another pathway.
No, we haven't. And actually, to be honest, this is because it's quite, we've got these results from different studies, I think, from different cohorts pointing always there. This is something we need to sit down and discuss and see more, the pathway in focus. So far it was like starting agnostically from looking all of the metabolites, and actually our result was always point to that. But I think this is something we really need to sit down and look at different enzymes, as you say. Yes, no, we haven't done it, but it would be great. Yeah.
And all of these PUFA levels, polyunsaturated fatty acids, have been actually included in specific phospholipids or in other lipid classes. For example, have you measured other lipid classes, such as sphingolipids or?
So again, in some of the studies, yes. So in the paper that I maybe would have been very interesting for me to have added, we had looked through mass spec different phospholipids and with different amount of saturation and different fatty acids, and then we saw differences in that. In the Nightingale data, you cannot see that. It just gives you, the fatty acids, polyunsaturated fatty acid, and then it just gives you one molecule, one sphingomyelin. But in the lipidomics analysis we've done, we have looked into that, yes, and we do see differences between, different fatty acid saturation and chains, and in sphingolipids, phosphatidylcholins, and so on. Yeah.
Okay. Very nice. Very nice.
Great discussion, guys.
We do have some data from our local studies on lipidomics, and if there's any projects you have in mind, we'd be very happy to collaborate and share. They all have, they are well, they've got lipidomics. Some of them have NMR as well. They have genetics. Yeah.
Okay.
I'm smelling the first collaboration. That's great. That's the spirit of this Bridges seminar. Great. And I'm sure people will think about your work, Petra, which is really great. And thanks again for being with us today. Sorry for keeping you a bit later because we're late today. I don't know. Thanks again. And we'll hope to see you soon in Greece to give us a live talk. For example, next meeting of Genedis, which will be in October, as far as I remember. I'm sure people will invite you. November, sorry. So I have almost dementia, as usual. Thanks a lot. Have a nice afternoon.
Thank you so much.
Thank you, Petra.
For the invite.
Goodbye.
Have a lovely evening.
Good afternoon. See you next month to everybody else. Thank you.